Hello, I am Toby Ven, and I will be presenting my experiment that I executed for the Bio Expo. So I researched if peripheral blood molecular cells, most commonly known as PBMCs, are an indication of breast cancer. So before diving in what I researched about, I will be talking about how I got introduced to this project. Then in this presentation, I will be talking about my purpose for this research experiment. I will be then talking about what PBMCs are and why they are significant in my project. I will also be talking about the steps I took in figuring out my purpose. I will then present my data in both a table and a graph. And lastly, I will draw conclusions from my data. So how did I get interested in this project? Well, while I was reading around, I stumbled upon an article about how clinical pathologists in laboratory medicine use PBMC data to indicate liquid tumor cancers, most notably leukemia and lymphoma. I then wondered if it was possible to use this technique for solid tumors because the only way I knew solid tumors were diagnosed were through biopsies. This is what led me to the Fred Hutchinson Cancer Center, which then led me to Yuri Rubina. He has been a great help in this project and guided me throughout this experiment. So now let me dive into my project. So why did I do this experiment? I did this experiment to test if PBMC levels are an indication of solid tumor cancers. After my interest in seeing that PBMC are an indication of liquid tumors, I just got interested in testing this. If PBMC levels were an indication of solid tumors, then I wanted to also test if these levels could predict whether these solid cancers were malignant or benign. So the reason for trying to figure this question is so that cancer can be seen through blood rather than biopsies, which can be dangerous, or scans, which cannot tell if the tumor is benign or malignant. So what are PBMCs? PBMC stands for peripheral blood molecular cells, which are blood cells that have a round nucleus. These round nucleus blood cells function as infection fighters. So when there is an infection, the levels of the blood cells will increase as there are more cells needed to fight off the infection. So I'm using PBMCs to try and indicate cancer because this could be a safer way for diagnosing cancer rather than doing a biopsy. I am primarily focusing on breast cancer and using white blood cells as a type of PBMC. So the first steps I took for my project were blood collection and PBMC preparation. I did not collect the blood as that was done prior, but I did prepare some trypan blue for the hemocytometer. I put the sample in the hemocytometer and counted the cells manually. Then I checked the count with the Nexcelom cellometer, which is automated. So this Nexcelom cellometer was used to check my manual counting so that I got it right. We then allocated the liquids, which means that we transferred the liquids to containers. And here are my results for the experiment. So this table shows when they were born, the gene data, if they had an MRI, if the patient was biopsy eligible, which was graded on the BIRAD system. It also contains if the patient had a biopsy, it shows if the tumor was malignant or benign. This table also includes the most important part, the white blood cell, measured in thousands per microliters. And then this table also contains previous history of cancer and previous family history of cancer. And lastly, it contains the Nexcelom cell count, which was a check on the white blood cell counts counted manually from the hemocytometer. So I recorded the gene data in the family history of cancer for further investigation if the PBMC was an indication of cancer. Well, here is a graph of the results, and as you can see, there is no correlation of white blood counts with patients having cancer. These white blood counts are a type of PBMC. They were all between 4,000 and 8,000 cells per microliters, which is the normal range for humans without having cancer. All these patients had cancer though, so there are no correlations between white blood cell counts and having a tumor, which means that there's no way that there is a correlation between benign tumors or malignant tumors with white blood counts. But you can see yourself that there's no correlation, as benign tumors marked in blue and malignant tumors marked in red show no correlation with the white blood counts, even though they are different types of cancer. So what I concluded from this experiment was that there was no correlation between PBMC levels and cancer, which means that there was no correlation between white blood cell counts and malignant solid tumors, as there is no correlation between any kind of cancer or tumors with PBMC levels. But we can see that white blood cell levels are an indication of liquid tumors. We already knew that going into this experiment, and that was stated before in this presentation, and that is why I was interested in doing this experiment. So it was shown in this data too, because the outlier shown in the graph earlier, it was someone who had leukemia. And so that's the only thing we can really conclude from this experiment. But from this experiment, I have learned a lot, because I learned how to conduct an experiment and also doing some lab work. Well, that's all I have to present. And thank you so much for listening, and have a good day.